I'm happy and I'm really excited to share with you our first experience with TruPath Genomes. And I want to share the experience, but also the perspective that we probably see in the near future for TruPath Genomes. My name is Marcel Nelen. I work as a laboratory specialist in clinical genetics, and I'm also heading the genome diagnostic section in Utrecht, in the Netherlands. This is my conflict of interest slide. There's not much to disclose, so I'll skip that quickly. Just to give you a little bit of background where we're coming from, the genome diagnostic section is located in the city of Utrecht, which is right at the center of the Netherlands. We are one of the 7 university medical centers in the Netherlands. And to be able to do genetic testing in the Netherlands, you have to have a permit that is given by the Ministry of Health. So there's actually no commercial competition in the Netherlands that is allowed to do genetic testing. We serve about 2.5 million individuals in our care area, and it's more or less the same for all university medical centers. In 2010, we started to use next-gen sequencing, and in 2013, we switched to whole exome sequencing, and that again has changed last year when we started to use whole genome sequencing for our intellectual disability patients. This year, we hope to transition all of our clinical diagnostic workup to short-read whole genome sequencing, and then hopefully we'll make a switch in the near future to TruPath Genomes. This is the overall goal. What we're working towards is to have a single, robust workflow that actually fits all of our needs and basically can answer most of the questions that come from the clinic. For that, we have now developed a workflow and we have it in place in the lab for short-read whole genome sequencing, and that is divided over 4 different modules. The first module deals with normalization and quantification of DNA. The second module is used for whole genome sequencing library preparation. For that, we use the Volta system Callisto. And in module 3, of course, the sequencing takes place on the NovaSeq X Plus. And of course, also the secondary analysis is done. And for that, we use the ICA environment of Illumina and all the Dragon pipelines that comes with that. The fourth module deals with the VCFs and the BAM files that are generated in module 3. And for that, we use the Franklin informatics platform, and we started to use that last year. We use that for variant annotation and, of course, also variant classification. Are we there yet? So do we have a workflow that is fitting most of our needs? Not yet, but I think TruPath Genomes promises improvements on certain areas that we think we still have additional tests to do in the lab. So, and Illumina, when they approached us to be alpha testing the TruPath Genomes, promised us to show improvements on difficult regions, to show improvement in phasing, and to show also improvement in structural variant detection. I will show examples of all these 3 topics, and hopefully I can convince you that Illumina actually delivered on their promise. So first, let me start by discussing difficult regions, and one of the most difficult regions that I know of is the region that is involved in spinal muscular atrophy. Depicted on the left, you see a cartoon of a family that was identified in our HealStick program. And one of the difficult cases in doing diagnostics in this particular case is that the father is a silent carrier. Difficult to recognize, and for that we use currently the Asuragen kit, which is PCR based. And it is capable of detecting these silent carriers, but it's difficult, and we also need confirmatory testing to actually show that this is the case. The trio, the family that you see here in this plot is actually published, and we published on the data that we got from PacBio sequencing and ONT sequencing. It turned out to be quite a difficult case, and it took us several months to actually crack it. And what you see is that the mother is carrying 2 SMN1 copies and 2 SMN2 copies. The father is actually also carrying 2 SMN1 copy, but it's on 1 haplotype. The other haplotype doesn't carry an SMN1 copy, but has 3 copies of SMN2. The child has a de novo mutation on the allele that she inherited from the mother, and also inherited from the father an allele that doesn't have an SMN1 copy. Hence, she has developed a spinal muscular atrophy. And we were thinking that, well, this is a perfect case to test TruPath genomes. And in the gray box, you actually see the data that we got from the TruPath genomes from the father. If we would have used just regular standard whole genome sequencing, we would only get the copy number information. And that's actually what you see in the top part of this gray box. You see that the total copy number is 6, 2 copies for SMN1, and 4 copies for SMN2. What happens is that normally it would stop here. You don't get information on phasing. But actually, in the red box, it is depicted the additional information that TruPath genomes is giving us. It shows that there is an SMN1 duplication. It shows with high confidence, and I think this is a Phred score, that this duplication is actually true. And it shows that there is a duplication filter pass that shows that the duplication is actually on 1 haplotype. So using TruPath genomes and the newly developed Dragon MRJD pipeline, and of course the targeted approach, we were able to confirm the data that we received from ONT and PacBio data and show that the father is actually a silent carrier. The next case is a bit more difficult and is basically dealing with phasing. What we currently see here is a self-designed algorithm that we use to stratify the phasing data that we get from the TruPath genomes. What we actually do is we sequenced a trio, so the father, the mother, and the child, and so we have a true set of SNP calling, and we compare that to the phase blocks that the TruPath genome is actually giving to us. You see here a region that is quite interesting for us because it's one of the most frequently asked genes for NIPD. And phasing in NIPD is actually critical to have that correct. And what you see here is a region of 1.4 megabases. And I will talk you through the colors and the dots that you see. What you actually see in the lower part of the slide is DNA isolation using just standard protocols. The top part of this cartoon is actually showing data where we use high molecular weight DNA isolation protocols. You can clearly see the advantage. It's less noisy. It's much more accurate. The green dots are actually correct calls if you compare the phasing information from TruPath genomes to the true set that we developed. If you move towards the middle of the two haplotypes, then you see that there is a color change. And that color change actually indicates that the SNP call is less reliable, and the lines in between actually indicate that there is a possible switch in the haplotype. In the lower part, you see 3 separate phasing blocks. In the top part, you see just one phase block of 1.4 megabases, which to us was quite astonishing that we could achieve this type of information using a short-read whole genome sequencing. The next example that I want to show you is an example of an intron 22 inversion in the factor VIII gene, a gene that is involved in hemophilia A. And in the plots that you see, we show the inversion in a female carrier. What you see on the left is, again, the developed algorithm that our bioinformatics team designed. The same as the phasing plots that you show, but now the lines are arches, and that is basically to show that the SNPs that are connected over the distance that the arches breach. The top and the lower are just female controls. The middle part is the actual patient that we sequenced using TruPath genomes. We grayed out the green color just for clarification, but the blue dots are actually the factor VIII gene, where the factor VIII gene is located. And clearly you can see the long distance and the arches that are connected where the inversion took place. The right plot, again, is the co-location plot. And you see these two butterfly bow tie type of structures, and these are actually the sequences that indicate that the inversion took place. If you look carefully to these bow tie plots, you can actually distinguish if a true, let's say, a standard inversion took place, or if there are smaller deletions. The two dots should be together. If there is just a normal inversion, but you can clearly see that there is some sequencing missing. And I want to showcase that in the next example, where we show a more complex structural variation in the SDHA gene. The SDHA gene is involved in a rare tumor cancer syndrome. And it's a tumor suppressor gene. The plot on the left is actually the inversion deletion that you can see. The plot in the middle is a control. And you can actually dissect what happened in this structural variation. If you plot the lines connecting these bow tie structures, you can actually dissect the deletion on a base pair level. You can actually show the inversion on a base pair level. So if you do the math and do all the calculations, you can see that A2 is connected to B2, B2 is connected to B1, and B1 is connected to D1. The region in C is actually the deleted section of the gene. And if you look closely to the picture, you can actually see that the intensity of the plot is less. So coming to my concluding slide and my summary, TruPath Genomes is one step closer to a one-test-fits-all, in my opinion. The workflow is straightforward, very easy to use, and quick, rapid implementation is feasible. In essence, you still get a short-read whole genomes, but now with the add-ons of phasing information, improved performance, but also a novel approach to resolve structural variation. The capacity is 16 genomes per run, and hopefully the roadmap that Illumina is showing indicates that there will be a substantial increase if indexing becomes available. Clear visualization of structural variation I've shown. And what happened actually is now that we removed more or less module 2 from our standard infrastructure. We have now one module for DNA isolation of high molecular weight. We have sequencing capacity using the Illumina Kinetic Analytics and the Dragon pipelines, and we still have module 3 for variant annotation and classification. And with that, I'd like to conclude and acknowledge all the people that are involved in this alpha testing project, and of course also Illumina for allowing us to do this alpha testing.